Hi everyone, I'm Nézet, and I will present you for the first time today my work on the new NF core pipeline phase impute dedicated to genetic imputation. But before presenting the pipeline in itself, I would like to explain you why genetic imputation is useful and how it works. As many of now that the Nextflow community is growing, some of us are not necessarily familiar with the genetics and how it works. So genetic studies aims to understand how genetic variation influences traits, but also disease. To do so, the first thing we need is reliable and as comprehensive as possible DNA information. When using sequencing techniques, we get a sequence of nucleotides called reads that we align to a reference genome. We then identify the difference between the target DNA information and the reference genome by calling the variants. As you may see, the genetic variation is noted as a combination of two nucleotides called allele, and indeed, humans, as well as many other species, are diploid, meaning that they possess two sets for each chromosome. One is inherited from the father, and the other one from the mother. Therefore, they aren't completely identical. As a consequence, we need to distinguish between a genotype, a combination of allele at a given position, and a haplotype, the sequence of allele along a given chromosome. When studying a target DNA information, we may miss or not be highly confident in the genotype observed. Phasing and imputation are statistical analysis that helps to overcome this problem. Phasing aims to align alleles into haplotypes. We can then distinguish which allele belongs to each parental chromosome. This employs a hidden Markov model which considers the probability of recombination event and point mutation alongside the chromosome. Imputation algorithm, on the other hand, use statistical model to predict the likely unobserved alleles. But the phasing and imputation software, the use of them will be dependent on the data in input. It exists effectively multiple technology to extract the DNA information from biological samples, with a balance between the quantity of information we get, its quality, and its cost. High coverage whole genome sequencing provides the most comprehensive data by sequencing an individual's entire genome with a high number of reads covering each position. It allows to be highly confident in the genotype observed, but its downside, it's quite expensive. Genotyping, on the other hand, focuses on specific markers of interest, providing a cost-effective and targeted approach. Low-pass sequencing is a balance between the two. It sequences an individual's genome at a lower coverage, reducing the cost, but still provide more denser information than genotyping. Phasing and imputation analysis enable bioinformaticians to get the most out of their data. For high-coverage data, phasing allows to study the haplotype context of variants and hence its consequences on disease. Phasing shines even more when used with imputation on sparse data to complete the missing genotype. More specifically, for low-pass sequencing, it allows to still detect new variants of interest and then to uncover rare causal variants for disease at a low cost. To help people to use phasing and imputation software easily, I've decided with the help of Sol Pierotti and Annabella Trigila to create an NF core pipeline dedicated to genetic imputation named Phase Impute. The goal for the moment is to develop the following pipeline. It's a bit complex. It consists of multiple linked processes that I will now describe. The first one prepares the reference panel to be used for the next processes. The main step will be to phase it, if not already done, do some quality control, create specific software files, and separate each chromosome to chunk them in small bytes to parallelize the imputation process. This imputation process is the core of the pipeline. It is used to impute target individuals' files, BAM, CRAM, or even VCF, and to output the corresponding imputed VCF. Many different softwares already exist, and our aim is to make them all easily available in this pipeline. I'm helped on this task by Sol Pierotti for the Stitch software and Annabella Trigila for the Kilt. To ensure that the pipeline is working correctly, but also to perform accuracy analysis, I developed a simple simulation process to then sample a high coverage target file into either a genotype or a low-pass sequencing simulated file at different depths. This will be then imputed and compared to a truth VCF file in the concordance process. These steps will either use the concordance tools of Glimpse or a workflow in development by Sol. It generates concordance statistics that will help us to identify the more impactful parameters in the imputation process. Finally, a refinement process proposed by Sol will allow to filter the best imputed position in the imputed VCF and rerun the imputation process to improve the accuracy. The data needed to run this pipeline are the following: the target DNA information, some reference genome file, the reference panel to be used for the imputation, as well as some additional file for specific softwares. For the concordance analysis, you will also need a file with the allele frequency and a truth VCF to be compared to the imputed one for each individual. The main challenges I faced so far while developing this pipeline are the multiple entry points due to the multiple processes. As a consequence, I need to configure multiple CSV input files and to correctly set up the succession of processes. Another nightmare that many other bioinformaticians face in genetics is the presence or absence of the CHR prefix in multiple files. I will need to choose one configuration while aiming to reduce the duplication of file. As an example, I would like now to present you some preliminary results we have on human data with a collaboration with Anthony Herzig from the laboratory of Emmanuel Gemnin from the INSERM in Brest. We performed concordance analysis on 16 French individuals using the version 37 of the human genome. To do so, we used the 1000 genome reference panel phased, but also this same panel in addition to 800 French individuals. We simulated 4 different depths, studied the longest and shortest chromosome with or without a genetic map. For the allele frequency, we use the reference panel, and we compute the truth VCF for each individual using BCF tools and pileup. Concerning the imputation tools used for this analysis, we only use Glimpse 1 for the moment. Here are some of the results. On the x-axis is represented the minor allele frequency in percentage, and on the y-axis is the imputation accuracy from 0 to 100%. As expected, imputation accuracy tends to decrease as the allele frequency becomes less common and as the level of coverage decreases. Here, it's a bit more complicated, but it's exactly the same. It's compared here the chromosome 1 in light color and the chromosome 21 in dark color. The size of the chromosome doesn't seem to have a huge impact on the imputation quality, except maybe for lower coverage or rare variant. Similarly, the presence of a genetic map doesn't seem to improve a lot the imputation accuracy. More interestingly, on the other hand, the addition of individuals with a shared genetic background to the target DNA target DNA individuals inside the reference panel seems to impact positively the imputation accuracy. Moreover, this effect seems to be even more pronounced for rare variants. In conclusion, the phase impute NF-core pipeline will enable bioinformaticians to easily use imputation software and to test their accuracy. The first results are encouraging, but need to be expanded to other imputation tools and other configurations. I aim to use this pipeline for my PhD thesis to identify genes involved in canine dysplasia, a complex genetic disease, and to perform a comparative study with human data. I want to highly thank my team and our collaborator from the GGB team, the funders of my thesis, and of course the NF-core community, and more particularly Saul and Annabella for their contribution to the pipeline. Thanks for your attention, and I'm happy to answer any question.
Impressive work, thank you. So you have multiple pipelines involved there. How are you going to combine them? And specifically, are you planning to build a new mega pipeline concatenating them, or are you going to run them separately and sequentially? Does that make sense?
We were thinking for a time to add it to Sarek to make a meta pipeline with everything, but it would have been complicated. So the aim is really to have all those connected inside as presented in the global metro map. But yeah, for the moment, we want to stick on this plan, but I'm happy to improve. We are really at the start of the development of the pipeline, so we are open-minded on everything.
That's fair, that's fair. I think there's a balance to find between trying to get everything into one thing where you press the button and everything runs, and readability and usability, so good luck. It's going to be... Wait, hang on, don't go. I still have one more. How does the performance of the pipeline compare to web-based services for imputation, such as the Michigan imputation server?
Yeah. So that was one of the aim to use this, to create this pipeline is I'm working on dogs, and there isn't a lot of services allowing to use the new version of the dog's genome. And we asked to have the new panel available, but they asked for an astronomical cost to add the new version. So we wanted to have everything available easily and in open science. So for the moment, we don't have really performance. So the quality of the imputation is broadly the same as what we can find in the literature for the moment. But for the computational resources, I still have a lot of work to be done to compare to the available public services.
That's totally fair. And it will be a great resource for also other groups who want to run on other organisms, so extensible and all of that.
Yeah, that's the aim.
Yeah. Well, good luck. Thank you very much.
Thanks.